Assalamualaikum dear students, welcome to your channel The Anatomy Canvas. In today's class of histology of the foundation module, the lecture 2, we are going to discuss the cell histology, and in cell histology we will do the cytoskeleton of the cell, and we will discuss each type of the filaments in detail. So what is cytoskeleton? The cytoskeleton provides structural support to the cell, and the cytoskeleton also functions in cell motility and regulation. Cytoskeleton is a network of the fibers extending throughout the cytoplasm. The cytoskeleton organizes the structures and activities of the cell. So here you can see different types of the fibers and tubules which are forming the cytoskeleton, giving structural stability to the cell and also helping different functions of the cell. So there are the three main types of the fibers in the cytoskeleton. These are the microtubules, the microfilaments, and the intermediate filaments. As we know, we also classify the different parts of the cell as membranous and non-membranous organelles. So in non, we also include the microfilaments, microtubules, centrioles, and ribosomes in the non-membranous organelles as well. What is cytoskeleton? It is basically considered and bones and muscles of the cells. Very simple. It maintains the cell's shape and internal organization of the cell. It permits movement of the substances within the cell and movement of the external projections. Provides scaffold to keep the various organelles at their specific places. It facilitates the cytoplasmic streaming of the different molecules, the different subcellular organelles inside the cytoplasm. It will help in the motility of the cells, provide pseudopodia, also form part of the microvilli, also the microtubules forming the mitotic spindle, and centrioles also helping the cell during cell divisions. Different parts, different microfilaments are also helping different functions in cytoskeleton. So this is just a little bit introduction what the cytoskeleton is. So it is forming the core of the structures like microvilli. You can see these are the microvilli, these are the cell surface modification of the cell, and these microvilli are also formed by the cytoskeleton. The protein rods, filaments, and tubes running through the cytosol in different forms. They may be rods, they may be filaments, they may be in the form of the tubes which are running in the cell in different directions, and they maintain the shape of the cell and also maintain the changes in shape of the cell, and they also move the organelles inside the cytoplasm either from the periphery to the center or from the center to the periphery. So first of all, we will discuss the microfilaments. The microfilaments are the thinnest class of the cytoskeletal fibers. They are the solid rods of globular proteins. The actin about 5 nanometer in diameter. The actin microfilaments consist of twisted double chain of the actin subunits. So in this diagram you can see these are the intermediate filaments which are present. Here you can see the endoplasmic reticulum is present. Here you can see the microtubules are present, and in this area you can see the microfilaments are present. So these are the smallest of all, the thinnest class of the cytoskeletal fibers, having a diameter of usually 5 nanometer. They are formed by the actin molecules. They are designed to resist tension with other protein. They form a three-dimensional network just inside the plasma membrane. So here you can see these basically the actin protofilaments. They are the G-actin monomers are present, and these G-actin monomers polymerizes to form the double twisted helical structure of the actin filaments, also called the fibrillar or F-actin molecules, and these F-actin molecules then form the different other proteins which are derived from the F, like the filamin, the glycine, the spectrin, membrane, the cap stable filaments. So these are the different types of the proteins which are derived from the actin protein, and this actin protein is basically formed from the G-actin monomers. These G-actin monomer polymerizes to form the actin protofilaments. Also you can see at this in this diagram there this fibrillar actin molecules. They have a positive end or a growing end and a negative or minus end. So in positive end constantly the actin monomers are added, while at the negative end depolymerization is taking place and these monomers are removed from the negative end. So it is a dynamic structure. It maintains the structural stability. It helps the movement of the different molecules during the in the cytosol as well. In this diagram again you can see these are the different types of filament. One intermediate filament, other microtubules, and third is the microfilaments. So here you can see these are the microfilaments, and these are present in close relation to the plasma membrane over here. These are the tubular structures which are present over here. These are actually the microtubules, and in addition to this the intermediate filaments are also present. This is the electron micrographic picture of the different proteins, different filaments forming the cytoskeleton. Here you can see this is a diagrammatic presentation of the neuron, and the neuronal cell body is present over here. The nucleus, lysosomes, nucleolus, and the Golgi apparatus is present. And here you can see the axon, and axon terminal is there. And in this axon you can see the fibrillar proteins which are present, which are associated with this axon. There are different microtubules and other proteins which are present in it. So microtubule, these are the three different types of the protein. So far we are studying the actin protein, basically 5 to 8 nanometer in size. The microtubules having 25 nanometer in size, and the intermediate filament, these are tubular, basically having a diameter in between these two. That's why called intermediate. They have a diameter of 10 nanometer in size. And in this diagram you can see this one is the cell membrane. And this is the area also called the cell cortex, in which you can see the actin filaments are present. Here you can see these are the microtubules which are present. In this you can see this one is the mitochondria present. Here the microtubules are present. All these structures are the intermediate filaments, and here in this area you can see the microfilaments are present. So these are the three different types of the filamentous proteins and the microtubules which are forming the cytoskeleton in the cell. So microfilaments, they have the actin binding proteins. These proteins are very important because they help, these actin are basically acting as a motor. They are moving different materials from the inside the cytoplasm, from the periphery to the nucleus, or from the nucleus to the periphery, and for this purpose they are using specific actin binding proteins, and which will help them to move different different organelles or different molecules inside the cytosol. So these are present beneath the cell membrane in the microvilli, and also here you can see this is the structure of the microvilli, and beneath the microvilli, at the bottom of the microvilli, you can see the actin filaments are present, which are forming the web over here, also called the terminal web, and from here the actin microfilaments arises. These microfilaments also the actin, which are forming the core of the microvilli. They also anchor the other proteins in the cell cytoplasm, and they are also involved in forming the pseudopodia. For example, for the movement of the cell, like the WBCs, which are moving in the blood, and also in the connective tissue, different smaller cells which are moving. So these proteins basically help in formation of the pseudopodia, and they help the cells to move in different directions. In muscle cells, thousands of actin filaments are arranged parallel to one another. The thicker filaments composed of the motor protein myosin. So here the actin binding protein in muscle cell is myosin. It interdigitates with the thinner actin filaments. In this diagram you can see these are all the actin filaments which are present over here, and in these actin filaments you can see the myosin, these thicker proteins which are myosin are present. These are basically motor protein, also called the actin binding protein, and they help with the help of the actin and myosin the contraction takes place in the muscle cells. So myosin molecules walk along the actin filaments, pulling the stacks of the actin fibers together and shortening the cell. So next is the intermediate filaments. These intermediate filaments are a very diverse groups of the intermediate filaments, and these are basically organized into 6 major classes on the basis of the gene structure, the protein composition, and cellular distribution. So first and second class basically characterized as the keratins or the cytokeratin. They are most diverse type of the intermediate filaments. These are present in the stratified squamous keratinized type of epithelium, and in this epithelium the keratins or the cytokeratins are present in the form of the tonofilaments in the cytoplasm. The third class is the class 3, that is the vimentin, desmin, the glial fibrillary acidic protein, and the peripherins. So vimentin is basically present in different type of the connective tissues, and also and it is also present in the cells like in the mesenchymal origin, the fibroblasts, the macrophages, and the endothelial cells, while desmin is present basically in the smooth muscle cells, the cardiac muscle cells, and the skeletal muscle cells. The glial fibrillary acidic proteins is present in the neuroglial cells, and the third class, the class 4 is the neurofilaments, which are present in the axons of the neurons. The class 5 are the lamins, which are associated with the nuclear envelope, and class 6 are the protein, which is a lens specific group of the intermediate filaments, or the beaded filaments. These are also called the beaded filaments. So these are the different 6 classes of intermediate filaments, and also please remember the major function of these intermediate filament is to give structural support to the cell. This is a very important function. They maintain the structure of the cell. So their size is basically 8 to 10 nanometer in size, and they are specialized for bearing tension. The intermediate filaments are more permanent fixtures of cytoskeleton than the other 2 classes. They reinforce cell shape and fix the organelles location. This is very important function, and also your MCQ as well. So here in this diagram, this is electron microscopic picture. Here you can see these intermediate filaments are present in the cytosol, and these are the microfilament forming terminal web over here, and then these actin filaments going upwards to form the microvilli, the core of the microvilli. So this is electron microscopic picture. You must remember that intermediate filaments basically responsible for the structural support of the cell. So shape of the microvilli in the intestinal cells are supported by microfilaments anchored to a network of intermediate filaments. Here you can also see these are intermediate filaments. Here terminal web is present, and here the actin filaments forming core of the microvilli. So these microfilaments are attached to the intermediate filaments. These are anchored to the intermediate filaments. Next, the third class is the microtubules, and the microtubules basically these are the hollow cylinders having a outer diameter of the tubulars. These are tubular in structure, and outer diameter of these tubules is 25 nanometer, while inner diameter is 15 nanometer. They are made up of the tubulin molecules or tubulin dimers. The basic subunit is a heterodimer of alpha and beta tubulin. So here you can see the alpha and beta tubulin monomers, they combine to form alpha and beta dimers, and then they polymerizes to form the tubulin microtubules. So 13 protofilaments in a typical cylinder usually present. When you see the cross section, you can see usually 13 protofilaments will be present. Also, just like the microfilaments, they are also having a positive end, which is a fast growing end, and a negative end, which is a slow growing end. The GTP are very important in assembly. At the fast growing end, at the positive end, the tubulin dimers are attached to GTP, and this GTP provides energy for these cells for the movement of different substances across the microtubules. So alpha and beta monomers, you can see this is a basic structural unit of the microtubule. The heterodimers does not come apart once formed. The alpha and the beta tubulins are homologous but not identical. Each has a nucleotide binding site. So alpha tubulin has a bound GTP that does not hydrolyze, while beta tubulin may have a bound GTP or GTP. It varies. This GTP hydrolyzes to form GTP and gives the energy for the cell. Here you can see the microtubule. This one is the positive or fast growing end, and this one is the negative or the slow growing end. Here you can see the beta and alpha monomers having GTP, and here you can see beta and alpha monomers, the beta having GTP, because this GTP has been hydrolyzed to give energy, and it has become now the diphosphate. So microtubule is a hollow cylinder 24 nanometer in diameter. Along microtubule axis, tubulin heteromers join end to end to form the protofilaments with alternating alpha and beta subunits. So staggered assembly of the 13 protofilaments yields a helical arrangement of the tubulin heterodiameters in the cylinder wall. Here you can also see the alpha and the beta tubulin molecules are the forming the dimeric structures or the trimeric structure. So doublet and triplet microtubules. The wall of the one microtubule partly consists of wall of an attached microtubules. So in this you can see wall of the microtubule A is complete while that of the B is incomplete. It is a C shape and it shares this part with the A microtubule, and also in this case when it is formed by the triplet structure, the three monomers. So C is also incomplete, it is C shape, and it also shares a part of B monomer with it. So microtubules, again remember, these are thickest fibers. They are the hollow rods about 25 micrometer in diameter. The microtubule fibers are constructed of globular protein, the tubulin, and they grow or shrink as more molecules are added or removed. They move the chromosome during cell division. So you can see this one. This is the tubulin monomer. This is a microtubule, and attached to is the microtubule, the binding protein which is attached to it, and it has a receptor for the motor molecule, and organelle is attached to it. So in this way, these are the motor molecules which are attached to microtubules and which help the different organelles to move in the cytoplasm. So another function is as tracks that guide the motor proteins carrying organelles to their destination. The cytoskeletons play a major role in cell motility. This involves both changes in cell location and limited movements of parts of the cell. The cytoskeleton interact with the motor proteins. In cilia and flagella, motor proteins pull the components of the cytoskeleton past each other. This is true in muscle cells. So in this diagram, you can see these are the microtubules of the cytoskeleton. This is another microtubule of the cytoskeleton, and these are the motor molecules where you can see ATP is required to provide energy, and when this ATP is utilized, ATP is powered, and this is the way how these microtubules can shrink or can change the shape or can help in motility of the cell. So motor molecules also carry vesicles or organelles to various destinations along the monorails provided by the cytoskeleton. So this is acting as a monorail, and this one is the motor molecule to which the organelle is attached. Interactions the motor proteins and cytoskeleton circulates the material within the cell via streaming. So this is a beautiful diagram in which you can see these are the tubulin microtubules which are form of the tubulin dimers, and here you can see different motor molecules which are acting as a cargo. So this one is the kinesin and this one is the dynein. These are moving the substances. The dyneins are acting as a motor molecule like lysosomes or the mitochondria, and these are moving these organelles towards the negative end, while kinesin is moving in opposite direction like endocytotic vesicles are attached to the cargo receptor, that is kinesin. It moves on the microtubules towards the positive end and also moves the organelles from the periphery to the area of the nucleus, and dyneins move the organelles from the nucleus towards the periphery. So these two motor molecules, the kinesins and the dyneins, they are moving the organelles opposite to each other. So the functions of these different microfilaments and microtubules are that they give mechanical support, they maintain the shape, they stabilizes and balance the opposing forces, provide anchorage for organelles, they are dynamic, they dismantle in one spot and reassemble in another to change the cell shape. So this is another beautiful diagram to show the structure of the different parts of the cytoskeleton. First, you can see these are the microvilli, and microvilli, the core of the microvillus is formed by the actin, which is microfilament, and at the base of the microvilli you can see the terminal web is present, which is also formed by the actin. This terminal web is anchored by the intermediate type of the filaments, which gives attachment and anchorage to the microvillus. Then you can see the intermediate type of the filaments which are present, which give the structural support, and then you can see the microtubules are also present, which are acting as the cargo, the motors. These are the microtubules which are either moving the different parts from one direction to another direction. In this you can see the intracellular transport. Here you can see the motor proteins which are attached to the intermediate filaments, the spatial organization of the actin filaments, and here you can see the motor proteins, the actin filaments are attached to them. So the fact that the microtubules are not fixed means that the cell can remodel their shape, like plant cells or our cells in mitosis round up as the microtubules used to make the spindle fibers. So in many cells, microtubules grow out from the centrosome near the nucleus. These microtubules resist compression to the cells. So here you can see the differences between the three different types of the structures forming the cytoskeleton, the first microtubule, then the microfilaments and intermediate. So the property is the structure that the microtubules are hollow tubes. Wall consists of 13 columns of tubulin molecules. Here you can see the microfilaments, the two intertwined strands of the actin filaments. Intermediate filament, the fibrous proteins supercoiled into the thicker cables. Diameter is 25 nanometer with the 15 nanometer from the inner aspect. Here the diameter is 7 nanometer and here it is 8 to 12. The protein subunit here are the tubulin consisting of alpha and beta. Here the proteins are the actin, and here very different type of the diversity of the proteins are present in this group. So main function is maintenance of the cell shape, compression and resisting girders, the cell motility in the cilia and the flagella, the chromosome movements in cell division and organelles movement. These are the function of the microtubules. In case of the microfilaments, the maintenance of the cell shape, that is tension bearing, change in cell shape like muscle contraction, cytoplasmic streaming, cell motility and helping in the cell division. Then the intermediate filaments, they maintain the cell shape, anchorage of the nucleus and certain other organelles and formation of the nuclear lamina. So now we move on to the centrioles. Centrioles generally appear in animal cells. They look like two cylinders at right angle to one another. When viewed with an electron microscope, the cylinders show up as 9 bundles of tiny microtubules arranged in a circle. They help to form the fibers that move chromosome around when the cell is dividing. And as animal cells prepare for cell division, these two centrioles separate and go to the opposite ends of the cells. So here you can see these two centrioles which are present at right angle to each other, and in the area which is called the centrosome, where these are present, they have a cartwheel structure at one end. So the centrosome is a mass of the protein, also called microtubule organizing center or pericentral material surround the centriole in animal cell. So the microtubule organizing center is basically the center where the centrioles are basically present, and this is the area where large number of the microtubules are present. The alpha and beta tubulin dimers are present, which are required to form the centrioles. So following duplication, the pericentriolar material initially is associated only with each parent centriolar cylinder. So here you can see these are the mitotic spindles which are formed by the microtubules. So centrosomes are present, and here you can see the astral microtubule, the chromosomal microtubules, the polar microtubules. These are all formed from the microtubules of the centrioles. So centrioles are basically clusters of the microtubules usually paired near the nucleus. They organize the mitotic spindles. They are bases, that is motors for the cilia and flagella. And here you can see these are present in the form of a triplet of the in a structure of triplet of 9 structures, in which you can see the 3 monomers, the alpha, beta and AB, and C3 triplet of 3 are formed by AB and C monomers. 9 into 3 microtubule structures, 2 centrioles, cell center these are the beta tubulin. These are present at right angle to each other, and their function is formation of the mitotic spindles, both in mitosis and meiosis, and also the flagella, the basal bodies and the cilia. So in animal cells, the centrosome has a pair of centriole, each with the 9 triplets of microtubules. Here you can see these 9 triplets of microtubules. In this diagram, you can see the cross section, and in this diagram, you can see the longitudinal section. So during cell division, just before cell division, in the S phase, they also replicate. They have the microtubules associated protein that influence their linking them together, stabilizing them or destabilizing them. They form a network coming from the microtubules organizing center, which is usually the centrosome or the centriole. That is the negative anchored here. Also form the cilia and the flagella and the spindle fibers in the cells. So here you can see triplet of 9 structure. This is cross section, and this one is the longitudinal section. Now you can see the structure of the cilium or the cilia, short whip-like appendage, move material past cell lining of trachea and bronchi. They are similar to the flagellum, the longer on the sperm. At the basis of the cilia, the basal bodies are present, and these basal bodies are derived from the centrioles, and they form the structure of the cilia. So in this diagram, you can see this is the structure of the cilia. In the center, you can see the 2 microtubules are present, and at the periphery, these are formed by the doublets of the 9. So you can see the 1 microtubule, the second microtubule, and to these microtubule, the dynein arms are present, which help them to stabilize. These are the outer dynein arms, and these are the inner dynein arm, and these one are the interdoublets and nexine bridges, which are attaching the different monomers to each other. This one is the central sheath of the this cilium, and this one A tubule and the B tubule, which is present. So cilia are the short, and many flagella is long, usually one, few not the same as the bacterial flagellum. So here you can see the structure of the cilia, which A and B microtubules are forming the structures, and here you can see the outer dinine arms having a diameter of 24 nanometer. Here you can see the radial spoke is present, and these all structures help in it is forming the structure of the cilia, and cilia move, and they help to move the substances which are present in the lumen of the trachea and bronchi. So here you can see in detail the structure of the cilium. This one is the outer doublet of the cilia, and here this one are the radial spokes which are present. This one is the central sheath, and these are the central microtubules which are present. So you must remember that in case of this is the cross section of the cilia, the cross section of the cilia in which you can see A and B tubules which are present, and in the center the center you can see A and B, the doublets which are present over here. So you must remember these 9 doublets which are present at the periphery, they arises from the basal body, which also arises itself from the centriole, while this the 2 tubules which are present in the center, they arises de novo. So microtubules are the central structural support in cilia and flagella. Both can move unicellular and small multicellular organisms by propelling the water past the organism. If these structures are anchored in a large structure, they move the fluid over the surface. For example, cilia sweeps mucus carrying tapered debris from the lungs. So you can see this one is the electron micrographic picture taken from the epithelium of the trachea and the bronchi, and in which you can see these are the cilia which are present. They help the movement of the cilia. The substance which is present in the lumen of the trachea is bronchi is moved towards the oral cavity to be expelled, and this is the also you can see the section of the epithelium. This is the apical surface. Here you can see the cilia which are present over here. Cilia usually occur in large number on the cell surface. They are about 0.25 microns in diameter and 2 to 20 microns long. They are usually just one or few flagella per cell, and the flagella are same width as the cilia but 10 to 20 microns meter long. So the length is more in case of the flagella as compared to cilia. Flagellum has an undulatory movement. Force is generated parallel to the flagellum axis. In this is the section of this is the diagram of the spermatozoa, and this one is the direction of the swimming of the spermatozoa. Cilia move more like oars with alternating powers and recovery stroke. They generate force perpendicular to the ciliary axis. So in this you can see this is the direction of the active stroke and direction of the recovery stroke. In spite of their difference, both cilia and flagella have the same ultrastructure. Both have a core of microtubule sheathed by plasma membrane. 9 doublets of microtubule arranged around a pair at the center 9+2 pattern. The flexible veins of protein connect outer doublets to each other and to the core. The outer doublets are also connected by motor proteins. The cilium or the flagella is anchored in the cell by basal body, whose structure is identical to the centriole, and it is also derived from the centriole. So here you can see this is the cross section of the cilia. This one is the outer microtubule doublet. These are the 2 inner microtubules which are present. These are the smaller dinine arms, and these are the radial spokes which are attaching the inner doublets to the outer microtubules doublets. So bending of the cilia and the flagella is driven by the arms of the motor protein, that is the dinine arms. Addition to the dinine of the phosphate group form ATP, and its removal causes conformational changes in the protein. So dinine arms alternately grab, move and release the outer microtubules. The protein cross-links limit the sliding, and the force is expressed as bending. So in this you can see these are the microtubules which are present in the cilia, and these are the dinine arms. So dinine attachment to the dinine, addition of the ATP to the dinine, and then release help in the movement of these microtubules and movement of this cilia. So the references for the today's class are the Basic Histology by Siddiq, the Histology Text 10 is test by Jan Quera, and Histology Text a text and atlas by H. Ross. Thank you very much for your attention for today's class. Allah Hafiz till next class.